This is group 8 for the intermediate synthetic biology 2024 by synthetic SynBio4ALL Africa. We're here to present our project title, Acylase as potential quorum quencher in Pseudomonas aeruginosa. My name is Wachira. My fellow colleagues are Abijith, Esther, Hellen, and Joy.
What is antimicrobial resistance? This is the ability of a microbe to survive exposure to an antimicrobial agent that was previously an effective treatment. Reviews by WHO and Lord Jim O'Neill state that several millions of people will die due to AMR in the coming years. So this is one of the biggest global threats that we need to handle with alternative means. Next slide. This is the intrinsic mechanism of an antibiotic resistance in bacteria. We have 3 antibiotics, namely A, B, and C. We have A and B entering a bacteria through the porine protein, and one will be removed by efflux, and another one will join the penicillin binding protein that cannot synthesize the peptidoglycan layer of the bacteria. We have the last antibiotic C, which will not have an access to the outer membrane. Next slide. So Pseudomonas aeruginosa is one of opportunistic gram-negative bacterium that was listed in 2014 by WHO as the one of the high group, and it is resistant to most of the first antibiotic drugs. It mainly affects immunocompromised individuals and those with cystic fibrosis. So we chose Pseudomonas aeruginosa as our bacteria because it is a menace bacteria. Next slide. So we have the existing interaction between Pseudomonas aeruginosa with Staphylococcus aureus. We see the 2 bacteria can coexist and interact in a single plate, but we are witnessing the Pseudomonas aeruginosa being very competitive compared to Staphylococcus aureus. So we have other means of measuring characterization of coexisting interaction using gene expression, bacterial survival, and even a dosage of carbon sources. Next slide. Here we are seeing 2 pathways. One is a normal air pathway, and another one is a pathway infected by cystic fibrosis. We see in the normal air pathway, the CFTR protein pore is open, allowing the calcium ion to move out of the lung epithelial cell. But in the cystic fibrosis airway, it is thickened by the mucus, which clogs the CFTR, hence inhibiting the removal of calcium ion. This will in turn cause deterioration of pulmonary function and ultimately respiratory failure and even the death of a patient, especially when they are infected with Pseudomonas aeruginosa. Next slide.
So the next slide is about the quorum sensing. Quorum sensing is one of the mechanisms that the Pseudomonas aeruginosa uses as a virulence factor in causing diseases to humans. So using our quorum sensing mechanism, we were able to design and beat it against its mechanism. So quorum sensing is a key signaling mechanism where it uses the gene expression and it's based on the cell population density. So what makes it attractive for the novel antimicrobial? It is because it controls most of the virulence factor production, especially the biofilm, which makes the bacteria behave like a community and making it hard for it to be destroyed using the antibiotics. So the quorum sensing is largely, mediated largely by a molecule called the acetyl homosertrin lactone, also known as the AHL. And it has two well-known quorum sensing, thus the LAS and the RHL. So the AHL contains homoserine lactone moiety and the acyl chain. So you should make this acyl chain. You should remember it. Next slide, please. So here are the classes of the signaling molecules that the quorum sensing uses. And it uses the first, the cell impermeament molecules, where the cells are not allowed inside, it's extracellular. On the second picture, it is intracellular, where it enters inside the cell. And on the third, it attaches to the cell. Next slide, please. So using the acyl chain in the AHL as discussed previously in one of the signaling molecules of the quorum sensing, we were able to discover Acylase enzyme, which catalyzes the acetyl groups from various compounds. And we got the homologous genes that encode for Acylase as follows. And in this particular project, we used the PVDQ gene to encode for Acylase and the PVDA as the inducer promoter. The PVDA is associated with the pyridine biosynthesis, where the pyridine and the pyocinin is one of the two sidiophores which the Pseudomonas aeruginosa acquires in order for it to grow using the biofilm formation. Next slide, please. So below you can see an image of the degradation of the homoserine lactone molecules, whereby the upper represents the lactonase enzyme and the below represents the Acylase enzyme, where the AHL is broken down with the Acylase to fatty acids and the homosertine lactone. So here we have the degradation using the Acylase, and we prefer the Acylase because it is more specific to the substrate it catalyzes. Next slide.
So here we about this is the project methodology. We have 6 stages of methodology. The first one, we have the selection of chassis bacteria. As you all know, chassis bacteria is a very important aspect when it comes to synthetic biology. So we are going to use E. coli. And then our chassis bacteria will be able to target our specific tissue, that is the throat and skin. This is where Pseudomonas tends to reside. Then our chassis bacteria will have mobility capabilities, meaning it will be able to self-propel itself towards the tissues. Also, it will be able to sense the local environment, meaning the signals using the inducer promoter, which will be specifically used to target and to identify any trigger from the signals. Then our chassis bacteria will also be able to respond to HLS, that is what you are targeting in the Pseudomonas, and also be able to hydrolyze it. Then it will also be able to produce Acylase molecules using the gene that will be able to engineer into the genome. And also lastly, it will be able to produce externally detectable signals, which will be able to be triggered and measured using the reporter. So this is just a diagram of an overview of a gram-negative bacterial cell wall structure. You can see the bacteria there. You can see it has flagella for mobility. We also have the two membranes, the outer and inner membrane. We also have the peptidoglycan layer. So basically, this is just an overview and shows how a gram-negative bacteria tends to be more recessive and also resistant to antibiotics compared to gram-positive bacteria. So this is an overview of the cell wall structure for chassis bacteria. Then stage 2, this is where we'll engineer the chassis bacteria. So we'll use a plasmid. This plasmid is where we're going to engineer our gene of interest. Then we are going to use restriction enzymes to join to digest the gene of interest. Then we're going to use the ligases to join the sticky ends. Then after the plasmid is engineered, then we'll introduce it to the chassis bacteria genome for it to be expressed. Then this is stage 3. This is where we're going to trigger Acylase production. This is the brief overview and the clear overview of our chassis gene structure. You can see on the farthest end, we have the PVDA inducer promoter. We also have our gene of interest, the PVDQ. So a PVDA inducer promoter will be responsible to trigger any signal for pyovadin in the environment. So our PVDA inducer promoter will remain inactive unless the environment contains the pyovadin siderophore, which will be produced by the Pseudomonas aeruginosa. The reason why we are triggering pyovadin siderophore is because pyovadin tends to have a higher affinity to iron compared to the other siderophore present in Pseudomonas aeruginosa. So our gene of interest, you can see it together with the lac operon. And to the farthest end, you can see our terminator and also the kill switch. The main objective of our kill switch is to enable us to have a control of this setup. So our kill switch will help us to control the production of Acylase whatsoever in any point where it's required. That's why we have a genetic kill switch in this structure.
So the transformation of the chassis E. coli should be done for the plasmid to enter into the E. coli. So we have thought of using electroporation as a methodology wherein we'll add the plasmid DNA to the cells, E. coli cells, and then we'll pass an electric shock of around 15 kilovolts per centimeter gradient and 5 microseconds pulse to lead to small pores on the membrane, which will allow the plasmid intake. Then successfully the transfer of plasmid can happen into, the cells can be transformed with the plasmid. So to successfully express the Acylase, so as my friend Nelson pointed out, we are using pyovridin as the signal for the production of Acylase enzyme. So specifically pyovridin we have chosen because of the increased frequency of pyovridin produced by clinical isolates of Pseudomonas aeruginosa and cystic fibrosis patients. And once the engineered E. coli reaches the site of infection, it will be triggered to produce Acylase by using the mechanism that my friend pointed out. And finally, the Acylase will hydrolyze the amide bond present in the acyl homoserine lactones, which is produced by Pseudomonas as a part of its quorum sensing, and lead to a disruption of its biofilm. This will ensure that any other additional antibiotics that are given, which are usually given for cystic fibrosis patients in a hospital setting, will have the ability to act on the bacteria and kill it effectively. Next slide. So two very important things for a synthetic biology project are monitoring and optimization. So one way of monitoring the growth of our chassis bacteria is, as Nelson pointed out, is using a genetic kill switch, which will use a specific environmental trigger, either excess growth or excess Acylase production as a trigger for its own killing. And we can also incorporate a reporter gene or a GFP-like gene, which we can monitor at regular intervals through sputum samples or something to check if there is enough amount of growth of the chassis bacteria and if it needs any control. And next slide. For optimization, we can do metabolic engineering to ensure that the chassis E. coli focuses more on producing acylase rather than its growth, which will also incorporate itself with the genetic kill switch and control its growth. And we can also optimize the media by producing it even by pre-treating it with siderophores and maintaining proper pH levels and so on, with which are similar to the lung conditions, so that it will have an enhanced acylase production. Another option is directed evolution, where we can evolve our chassis bacteria in the presence of lung milieu so that it can produce effectively in vivo.
The biosafety concerns of our project include our chassis acquiring virulence genes from Pseudomonas aeruginosa through horizontal gene transfer, and we shall solve this by mutating the pili genes and the transformation capability of our chassis. The second one is the genome of our chassis acquiring mutations, and we shall solve that by using direct evolution to enable us to harness the mutations in a controlled manner. The third concern is the infinite growth of our chassis, and we shall solve that by using a kill switch. We shall engineer our chassis to be selectively and controllably eliminated from the system. The pros and cons of our project include, the pros include it's a targeted delivery. It is highly efficient in that there is a rapid onset of the therapeutic action of the solution, and then it is easy to optimize performance, plus also the economic benefits. It is also a broader use case. It can detect the problem, which is Pseudomonas aeruginosa, and destroy it. And there are minimal chances of resistance. It is more controlled in that it maintains predictable behavior, and it delivers consistent therapeutic results. It is also more tunable because it can be modified genetically and metabolically, and it is highly adaptable. The cons include the process. The bottom-up approach is extremely costly. The strain is slow, and it is more difficult to engineer. There are lower chances of success, and then what works in vitro may not work in vivo in the body. The competing technologies with our solution are the current technologies that are existent, and our solution can work alongside them. These include conventional antibiotics, like the third-line antibiotics for cystic fibrosis. Their weakness is that they are not novel solutions. They are expensive, and it takes a long time for them to be developed. The second current technology is the quorum sensing inhibitors, like lactonases. Their weakness is that they are non-targeted, and then they don't have, there is an absence of pharmacokinetics and pharmacodynamics. The third current technology is probiotics. These are the ones that protect the lung microbiota. Their weakness is that they are not explored, and there are contrasting results available in literature. These are the email addresses of our presenters and instructors for this project. These are the references that we use to get our information and data. Thank you so much.